that they have a chance to record. I need to switch now the display settings. No, thanks. That sounds good. More here? Or maybe you can better decide. 1, 2, 1, 2, 3. Yes? Okay. Thank you very much. So, good morning, everyone. Now, in this lecture, we want to dive in a component that we have so far ignored, into membrane proteins. You might wonder, okay, why to bother about membrane proteins at all? And the reason for that is that actually membranes contain quite a number of membrane proteins, a quite high number. So this is just like a cartoon. And if you think about the proteins in the cell, 30% of all of the proteins in the cell are actually membrane proteins, roughly 20, 30%. So it's quite an amount, a high amount that the cells has of proteins that are in connection of a membrane or even embedded in a membrane. And also from the pharmacological point of view, they are of highly interest. So most of the drugs that you are actually using are directed against membrane proteins. They interfere with the function of membrane proteins, and that allows basically either to modulate the function of our cells, to activate cells, or to shut down cells. So I think maybe that's a motivation enough today to dive a little bit into the world of membrane proteins. So what is so special about those? No? And if we take now basically here the membrane, you are already familiar, it's the bilayer formed by lipids. You see that membrane proteins can have quite different topologies. So membrane proteins can be integrated in a membrane, or membrane proteins can be attached to a membrane. So you can basically classify them in so-called integral membrane proteins and peripheral membrane proteins. You can further divide them because now these proteins that are really embedded in the membrane, they can also have different topology. You see here one membrane protein that spans the membrane once. That would be a membrane protein that spans the membrane multiple times. This would be also a membrane protein that spans the membrane multiple times, but it uses a different type of folding. So that would be so-called alpha helices, that would be beta sheets. And that gives a different structure to the proteins, right? So this is basically a very compact protein, and that would be here more something like a channel type of protein. No? And then you have basically here proteins that are attached, and this protein here, it's somehow in between. It's either embedded or attached. If it's embedded, then it's basically only on one layer, and we call those monotopic proteins. So you can basically classify now also these proteins in the way, how many basically sides do they see? This would be a monotopic protein, seeing one side, a bitopic or bitopic protein, seeing the two sides, and that would be a polytopic protein, right? And then you have membrane proteins here that are clearly attached. This would be a protein that's attached by a hydrophobic anchor, for instance, by a lipid or a fatty acid. That would be a protein that's attached to another protein in the membrane, and those you can normally easily remove. These monotopic proteins, they are in a type of in-between. If they are really embedded, then you cannot remove them. If they are more attached, then they would belong to this class. So they, depending on the type of protein, might either be more a type of integral membrane protein, or they might more behave like an attached protein. That's still also discussions among scientists. And that's for each of these monotopic proteins, you probably need to define this based, for instance, on the method that you use to test how fixed they are in the membrane. Okay, so this already gives, of course, quite a variety of possibilities how proteins can be basically fold, how do they function. You would typically basically here see this type of proteins. We talked already, they would maybe be able to form a type of channel, or they would be able to transmit information from one side to the other side. While those ones here are basically only located on one leaflet, and they can basically function on that side, but cannot transfer signals to the other side. Now, when you look basically around the membrane proteins, now you can also classify them to function. So they can do different functions like transporters. That means they move material from one side of the membrane to the other. You can basically group them into a group of proteins that would be receptors, that would basically receive a signal on one side of the membrane, and then they would undergo conformational changes that transmits the signal into the cell. Or you can have basically enzymes. So these are, from the chemical side, basically catalysts. So these are proteins that catalyze chemical reactions, right? And of course, that doesn't need to be a reaction on the inside. It can be also a reaction outside, depending where the catalytical center of this protein is. Other proteins can be anchor proteins. So they can be responsible to hold other proteins on the membrane. So that gives quite a diversity, and depending on basically their function, they can be localized in different type of the membranes. Now, the first question we could ask, how can it be that one class of molecules, proteins, can do all this function? No? Because you know maybe for yourself, you know that some things you can do very good, and other things you cannot do very good. But how can it be that in nature, one class of molecules can either work as a transporter, or can work as a receptor, or can work as an enzyme, or can work as an anchor? And to understand this, or to answer this question, we need to basically go into the structure of proteins. How are proteins built? And this is just a brief reminder that the proteins all are built of amino acids. So that's actually an amino acid chain. Amino acids that are linked via a peptide bond, which is an amide bond. And now you have basically here this principle of building blocks again. You have 20 building blocks that you can combine. And depending on which building block you combine in which order, you receive different structures. For instance, here, a type of alpha helix, a secondary structure. So the interactions of basically these peptide bonds and the side chains of the amino acids drive secondary structures and tertiary structures. And now we understand, because depending on what type of amino acid combination you have, the structure can be different. And with this, nature can create different functions. So it's again this principle of building blocks, units, like we had for the lipid world. Now only that you use 20 amino acids. For those that are maybe interested more in, you know, not only the Earth, but maybe the space. Maybe somewhere else on another planet, we might not use 20 amino acids, we might use more. And then maybe this life there looks different in terms of what type of proteins do exist and what type of functions these proteins have. Yeah? So that would be astrobiology. If you get fascinated about this, could be also something interesting later for you to go. Okay, now these proteins, when they have basically folded into this three-dimensional structure, they can also start to interact. Either they can interact with themselves or with others. So they can form tertiary structures. That would be then even higher assemblies, right? So it could be that basically I would be one unit, you would be another unit, you, and we could team up, and then we would form even a higher oligomeric structure. And that allows then, again, different functions to occur. Yeah. So now you see already that these membrane proteins, they have something special. And what is so special? They see two different environments, complete different environments. They see here on that side, they see water. On that side, they see water. And on this side, they see a very hydrophobic environment. So compared to a cytosolic protein that only is surrounded by water, these proteins have to deal with two different environments, two different, extreme different environments. And that's only possible because they, of course, use their amino acid composition to adapt to these environments. So those parts that are in the membrane, that will be hydrophobic parts in the protein also. They will be basically formed by a combination of amino acids that are hydrophobic. Those that are exposed to the two sides of the membrane, outside of the membrane, they will be composed of protein or amino acid combinations that are hydrophilic. Yeah? So for a membrane protein, it's actually, they need to have these two different environments. So if you want to work with them, if you want to take them out, it's difficult because you need somehow either to provide, again, a hydrophobic environment, which would be lipids, or something which can compensate for that. So if you want to work with them, there's only one way. You need so-called membrane mimics. You need to basically provide these membrane proteins with a possibility still to have a hydrophobic environment and a hydrophilic environment. And there's different ways to do this. You can do this, for instance, by using like detergents or like cone-shaped lipids that basically would stabilize these hydrophobic regions. You can, of course, integrate them in a bilayer again. Then they would be happy. Or you can use this type of bicell, so type of layer that is double layer that basically embeds the protein. You can put them in the vesicle, or you can put them in type of membrane sheet. And we will discuss a little bit more vesicles and also nanodiscs in these sessions here. Okay? So membrane proteins, to work with them, you need to provide them something like a compensation. If you take them out of a lipid bilayer, something like a compensation that looks like a lipid bilayer, at least has some of these hydrophobic properties in addition to the hydrophilic properties of the environment. Okay, so having now this means integral membrane proteins that are really embedded. That's the only way. You need to move them out and to move them in to a membrane-like environment. And the process that we are basically taking them out, we call solubilization. And you would need basically a detergent for that. It's like a soap molecule. So these molecules are basically this type of inverted cone shapes. So you have basically a very large head group, a small tail, and those molecules they would not form membranes. So if you add an excess and they start to integrate, they disintegrate the bilayer. They disintegrate simply the bilayer. So what you get is basically proteins with a bit lipid bound, with a little bit detergent bound. You get lipids surrounded by detergent. And now you have basically all the proteins freed out of the membrane. So they are in solution, but still have lipids and detergents on their hydrophobic part. That's the only way. And now you can basically add them to liposomes, or you can combine them with an excess of lipids and start to form proteoliposomes. That's a process that we call reconstitution. So you let form a membrane again, now containing the membrane protein, or you basically add them in these type of nanodiscs. For that you would basically not only need lipids, but also these helper proteins. The details we will discuss in one of the later sessions. Okay, so how do you do this practically? Practically, you would take cells, then you would, for instance, isolate the membrane that you are interested in. We have discussed this organelle isolation. So if you want to study mitochondrial protein, you isolate the mitochondria. If you want to study the Golgi, you isolate the Golgi apparatus. Now you have basically these organelles and cell membranes isolated. You add detergent, and you add basically more and more detergent that you completely disrupt the membranes, the lipids gets free, the proteins gets free, and then you have your solubilized protein. And if you are interested in a particular protein, you need to purify. And there's many ways of doing to purify proteins. Typically, you take advantage that these proteins have a property, a particular property, an affinity to something. If it's a charged protein, it would have, for instance, an affinity to a charged matrix where it can bind. Or if the protein is, for instance, modified with a little region where you have an antibody against, you can use antibodies to isolate the protein. Yeah? So there's different techniques. That's basically more, you go for the biochemists, they can help you how to purify the protein. But the principle is that you need to solubilize, and if you are interested in a particular protein, you need to purify this protein while you have it in solution. So the problem with this process is, actually you destroy the membrane. And I have pretended as if this is a process, no problem. Let's destroy the membrane and then you free the proteins. But some proteins are very sensitive if they are not anymore surrounded by their native lipid environment. So they might start to unfold, they might start to aggregate. So you lose the activity of this protein. So this can be a PhD, no? By itself. You can spend 3 years just to find the proper conditions to purify your protein, to actually solubilize and purify the protein. Because we have some ideas what is important, but for each membrane protein still we need to test this new. In many cases you are lucky. The protein that you are working on behaves like it basically is written in the book, but you can also be unlucky and you work on a membrane protein no one has worked before, and you need to find out the conditions that are optimal, right? And of course, you know, we remove some lipids, many lipids actually. We replace them by detergent molecules. That will have already an impact. Let's assume, for instance, we would form here a complex of 3 proteins that are normally working together. And now you start to remove lipids and you add detergent. You might disassemble. You would get me, you would get these 2 colleagues there. We would all be all 3 happy, but we would not anymore be functional because we have disrupted now the interaction between us that made us out, for instance, that was required for our particular function. Okay? And also detergent molecules by themselves, they can be harsh. You know, for instance, when you are using soap, there's also different type of soaps. No? If you have worked in the garden and you really want to clean, you might use a very strong soap, and you might soap very long. So if you soap the membrane proteins very long, you will remove all the lipids and replace completely with detergent. But for some membrane proteins, that would be too much. It's like with our own hands. If you overdo it, you actually damage even the skin. And you can imagine that detergents on proteins are also very harsh, and you can basically harm those. So you need basically to make a compromise between you want to get your protein very, free of lipids and nicely, single, as is isolated from the others, but you also want to keep the function. And that requires also always a compromise. Some lipids are even cofactors for proteins. So if you remove the lipids and you remove the cofactor, your protein is still there, but not functional anymore. So there's a lot of basically fine work required to, in the end, have your functional protein still preserved. Okay, let's have a look here. Here's basically a type of detergent. You see this idea. That's, it's sharps, it's not important. I just want to show you on this molecule, it has here a large head group and basically hydrophilic tail, and here the head group. Here's the head group and here's the hydrophilic tail. Typically, this is a large region, small region, and then basically these type of detergents, they would basically start to integrate in the membrane. They are not molecules that would form a bilayer. By adding an excess, they destroy the bilayer, and you basically get the proteins free. Okay, now we have basically purified here our proteins. Now we want to reconstitute them, so we want to bring them back in a lipid environment. And to do so, you would add lipids, an excess of lipids back. And here you can decide, now decide which lipids you want to use. Typically, if you want to get vesicles, the preferred structure of lipids would be the cylinder lipids, right, with these parameter P1 around. And they would then basically start to form vesicles. And that they would do in the moment you would basically remove the detergent excess. So you add the proteins in from the detergent with lipids, and then you start to remove the detergent. And then a miracle happens. You get this self-reorganization. What was the secret? Why would the lipids now, when you take out the detergent, self-assemble back? That was due to the particular properties of these lipids. They have always a hydrophilic part and a hydrophobic part. The hydrophobic parts want to interact with each other, but not with the water. So in the moment you take the detergent away, you get basically the reformation of the lipid membrane. And since you have also the membrane proteins there, you hope that they would also go in. There comes already a critical remark. There was, well, Thomas, is this really so? Well, for many vesicles it will be like that. But other vesicles might not contain. Some vesicles will contain 20 proteins. Other vesicles will only contain 1 protein. So you get actually a quite heterogeneous mixture. Actually, this picture here is not completely correct, right? You will get vesicles with many proteins, vesicles with 1 protein, or vesicles without any protein. Yeah? Because it's a spontaneous process, the lipids will reassemble, and eventually proteins around will be integrated or not. And that depends, of course, also how much lipid you add. If you add a large excess, you get a lot of empty vesicles. If you add basically a large excess of protein, then many vesicles will contain protein and also many copies of those. So that's a process you can play. Interestingly enough, and we have not fully understood this, it's basically, even when you use this detergent method, there will be always some empty vesicles. Even if you try as much as to optimize, typically up to 10–20% will be always empty. So we call this a heterogeneous vesicle population that you generate. But for many purposes, this is already enough. If you are only interested, for instance, in activity of a protein and whether an inhibitor inhibits your protein or not, this heterogeneous population of vesicles having some with proteins, some without, doesn't matter in the assay. For other purposes, it can become critical, right? Okay, so now there's different ways of removing this detergent, and that depends also of the type of detergent. We talked a little bit about this yesterday when there was this term critical micelle concentration. So what we are talking about is basically what concentration of molecules it's required that they start to aggregate. Okay? You can imagine if you take basically a soap molecule, only one soap molecule, and you put it in the ocean, it will be a monomer. There's no partner. If you take two and you put it in the ocean, they might never find. So they will be still as monomers. But the more you actually increase the detergent concentration, the more there will be a chance that they start to form structures. And for, and some detergents have already the ability at very low molecule concentration to form micelles. Others need very high concentrations. So if you have a detergent which needs very high concentrations and you put a lot of water in, you dilute it out, it will basically easily become a monomeric version again. So if you have basically like detergents with a very high CMC, those you can remove by dialysis. That's no problem. That's a process where you basically have your sample in a little bag that has pores and allows the detergent to leave, but not your vesicles, because the pores are very small, too small that vesicles can pass, but large enough that detergent molecules can pass. So you can dilute out your detergent into the buffer solution that you have around, and you can remove it. For those that have very small critical micelle concentration, for those that already at very low concentrations, you get micelles. They are very difficult to actually remove, but there is a trick. There's a particular beads, so-called polystyrene beads, that have very fine pores, that have basically the capability to bind your detergent. So if you add those to your lipid protein detergent mix, these detergents will specifically start to bind to these beads, and the sediment on the bottom, and you can remove basically the supernatant that contains your vesicles that have been formed. Okay? So the reconstitution is that you have your membrane proteins that are in detergent, you add lipids back, and you remove the detergent. As a consequence, you get the self-assembly, in this case of vesicles. Okay, there's another trick that you can do to get your protein back into the vesicle. You don't start just from detergent with proteins where you add lipids. This is like a soup. No? This would be like when you do in the kitchen. You have basically vegetables, potatoes there, and you put everything in. And then you cook and cook until you basically start to remove more and more water, and then you get basically a different type of soup than you would have just without cooking. What you can do here is basically you use already preformed vesicles. And how to make vesicles you have already learned yesterday. You learned, for instance, how to make these giant vesicles. And we will also later in the course train you how to make large unilaminar vesicles of 200 nanometers. Yesterday, I don't know, did you watch? Was it 5, 10 micrometer? Did you make a little competition? The group with the largest vesicles that I got. Rashini, what was the size you got on the vesicles? Something like 5 micrometer, 10 micrometer yesterday? Yeah, so not very large. So you can use those vesicles and you destabilize them with detergent. So you add slowly more and more detergent. What will happen is these detergent molecules, they will enter. More and more will enter. And at a certain moment, of course, you destroy the vesicle. It's completely gone. But you can decide now at which stage you want to add your membrane protein. You can basically partially destabilize and add your proteins that you have basically in detergent from the solubilization step. You can add them in the very late step of destabilization, shortly before the vesicles collapse, or you can add them at that stage, and that was what I explained before. You just have lipids, detergent, proteins, right? And depending on your membrane protein and depending on the lipid composition of your vesicle, it might be better to use these preformed vesicles at that stage, destabilized, at that stage, or at that stage. So how can you find out whether your vesicle is destabilized and how much is destabilized? So one technique is you use type of light scattering. Okay? So the vesicles, they will scatter light. And you will see this when we prepare our small vesicles or large unilaminar vesicles on Friday. You will see that when you look to the solution, it will be milky. It's a milky solution because the light that hits our vesicles will be scattered. Of course, when you completely solubilize, and the solution is clear, almost water clear. So you can measure basically the scattering. When you have the vesicle, the scattering is very high. When you have destroyed the vesicle, the scattering is very low. And now you can start to follow basically, okay, what is the scattering with my vesicles? What is the scattering when I add now more and more detergent? And then you will basically see that at a certain moment, the scattering starts to fall down. So you get on the point where actually all the vesicles are now having detergent molecules inserted, and the first ones also start basically maybe even to disappear. And you can basically continue until half of your vesicles is almost disappeared, until all vesicles are disappeared, and you only are left with detergents and vesicles. Detergents, lipid molecules, and your free protein. So at that curve here, you can decide, do I want to add basically my protein in this region? Do I want to add my protein here or here? After I have added my protein, then I need to remove the detergent again, and I come back to the formation of vesicles. Okay? So that's a typical experiment that you can do, and since when you want, for instance, want to use different lipids for making different type of vesicles, because you are, for instance, interested, does my protein requires a particular lipid composition to be functional, you would need for each lipid composition to make this titration curve, because the detergent insertion differs for the different lipid compositions, and you might require for one lipid type more detergent compared to another lipid type. Okay? So now you basically would have here your purified protein. You would, for instance, have these preformed vesicles. You would have detergents. You mix all of this. You make the detergent removal by biobeads. We often also use these gel filtration, and you end up here with these vesicles and with your proteins. At least this is what you hope. But how do you know that in the end you have vesicles with your protein inserted? So what you can do is you take these vesicles now, these proteoliposomes, and you basically give them to gel electrophoresis. Okay? So if you do gel electrophoresis, you detect proteins here. This is basically gel where we have now separated the liposomes containing protein and lipids. The lipids will basically run on the very bottom, but the proteins, depending on their size, will basically show a particular pattern. Here we have integrated one protein, so we only see one protein band, and we have basically taken samples at that step, at that step, at that step, and we follow. And what you see is basically in each step your protein is there, and even then in the end in your protein preparation and your liposome preparation it's there. In this case this protein was also fluorescently tagged, so we can also follow the fluorescent marker. That basically the protein is indeed in your liposomes. So that's something which basically I always recommend to do, because even after this reconstitution step, you need to prove that your protein is also there. This would be a typical, of course, assay when you do that you can use for small vesicles, large vesicles, giant vesicles. For the giant vesicles, you had one advantage. Remember, you can go to the microscope and see when you have a fluorescent protein. Do I see in the fluorescent microscope my nice giant vesicle, shiny, because my protein is in? If not, you would see these giant vesicles not in the fluorescent mode, but only in the light mode, right? Okay. So now basically the protein is there. Good. And you can of course also quantify, did I lose some protein on the process? And also how much protein do I really have in my vesicles, right? Because it is not like that you saying, okay, I add 10 nanogram of protein, and I end up with 10 nanogram of protein. Because in this step of reconstitution, you might lose protein. Some protein might stick to your tube and never make it into the lipid vesicle. Some protein, if you use biobeads, might stick to your biobeads and might never make it into the vesicles. No? And you also know that you get a lot of empty vesicles, so there's no guarantee basically that the amount of protein you have started with will also end up in your vesicles. But if you later want to make quantitative analysis, it's very important that you know the number of proteins, of course, that are in your sample. So to be absolutely sure basically whether your protein is really in the vesicles, you can do an additional trick. You can make a floating. What does it mean? You basically make again a density gradient, and you basically load your sample on the bottom in a high density. If your protein is free, not surrounded by lipids, and you would centrifuge, your protein will stay in the bottom. Lipids are light, so the lipids will go up. If the protein is embedded in lipids, it will also go up. So now you can basically take your sample, load it on the bottom of a gradient, and basically do the centrifugation, and everything what is lipid-like will float. And if proteins are associated with your lipids, they also will float. If you have, for instance, a protein that is here, again, fluorescently tagged, you can just measure by taking now all of these little fractions here that are listed here. You take basically from the top here, always fractions, 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 until you are on the bottom. And on the bottom, if you only add protein here on the bottom without lipids, the protein will stay on the bottom. If you reconstitute your protein, then you see basically your protein will actually be in the fractions where also your lipids are. So it has floated up. And now you have proven my protein is really embedded into the lipisome. In this case, the lipid signal, you can, for instance, use also a fluorescent lipid that has a tracer and follow this, or you make a lipid determination. From all these fractions that you have collected, you just determine whether they contain lipids. One would be if you work, for instance, with phospholipids, you can determine whether there is phosphate, and which fractions is how much phosphate. Yeah, that would be a phosphate determination. It depends on what you prefer. The fastest would be, of course, the fluorescent lipids, because then you only can measure the fluorescence of the protein, and you use another fluorescent, let's say, fluorophore for the lipids. One would be, for instance, here in this case, red, RFP fluorescence protein, the red fluorescent protein, and the lipids could be green, the marker lipids, and then you can follow this. That's up to you. It's not necessary to do it with fluorescent lipids. You can just make a lipid determination for each of these fractions. For instance, based, when you do this for phospholipids, which we are typically using, based on phosphate determination. Okay. So what you also can do, if you, for instance, have a fluorescent lipid, you can, fluorescent protein, you can determine now the insertion. Because now I told you, okay, they self-assemble. You mix lipids, you mix detergents and protein and detergent, and then you remove the detergent, they assemble, or you destabilize liposome, add your protein that you have basically in the detergent, and it will insert. But it will insert randomly. You can typically not control in which direction the protein is inserted. That's a disadvantage, right? Because nature is very precise. When nature inserts membrane proteins, they only have one topology. Those parts that would be to the cytosolic part are to the cytosolic part. Those parts that are to the outside or to the lumen of organelles will be on that side. Now, in the reconstitution process, we cannot really control this. People try it, of course. There's also tricks again, but there's no 100% control over this full process. So eventually, your protein might be the right side out or the wrong side out. And you want to basically maybe determine this, because you need to know this. It's a maybe important parameter. So what you can do, if you have, for instance, a fluorophore, you can take advantage of a quencher. So a quencher is basically a substance that when you add it, reduces the fluorescence. So there's no light coming out anymore, because this quencher is basically leading to an energy transfer that is then gives the energy in form from heat free, but not in form from light. So for instance, for this red fluorescent protein, it would be kappa. The more kappa you add, the more this red fluorescent protein will be quenched, will be not giving fluorescence. And that you can basically use, because if you have some proteins that have the fluorophore accessible to the kappa, they will be quenched, the others not. And you can basically see here for the quenching curves, that when you have basically kappa concentrations, the more you add, the more you get quenching. And in the end, here you end up for those vesicles, for instance, by 50, or for those vesicles here on the very bottom. And that gives you an indication. This is more a 50-50% distributed protein, and that would be more a protein that has everything accessible to the outside of the vesicle. And you see that might even differ between lipid compositions. So if you work with different lipid compositions, with different liposomes, and you want to reconstitute your protein, they might all differ in the way the protein is oriented. And that might affect also then quantification if you think about activity. If your protein has an activity, but it's basically in one vesicle type oriented 100% right side out, in the other vesicle population only 50-50, you need to take this into account. Yeah? But there are techniques for that. And there's more techniques. So what I have presented was here this quencher technique. What you can also use if you have, for instance, antibodies that you add, they would bind basically to a particular part of the protein, or you use a site-specific labeling. That means you have a particular functional group only on one side of the protein that you can modify with a reagent that is basically reactive to this. Then you can also quantify, or you use a process that we call proteases. So these are basically enzymes that eat up other proteins. But of course, we don't want to eat up completely our protein, but what we take advantage here is that those parts that are now exposed to the outside, they are easily accessible to the protease. Those parts that are embedded or in the inside are not accessible. So you would basically eat up here only a little bit of your protein, and this is an indication then the part that has been lost, what basically part has been exposed to the protease. Yeah? So that's just to give you an idea. You can go, if you want to read more about this, of course, to the publications. But just that you get an idea, we have different approaches to determine the sightness of a protein that we have embedded into the liposomes. Okay, now the next warning. There's all a lot of warning here today, right? Again, you think you add the lipids, and now you want to, let's say, reconstitute your membrane protein in a more complex lipid composition. You want to come more closer to the biological membrane. You not only want to have one lipid type, but you want to have 2, 3, or 4. What would you do? Probably like you did yesterday. You take the lipids, lipid A, lipid B, lipid C. They are all in chloroform. You mix them. Then you evaporate, and you have a nice lipid film of lipid A, lipid B, lipid C. Then we hydrate, we add buffer back, and then we hope that you get vesicles, right? That would contain A, B, and C in the same ratio. In biology, or general, in science, of course, this is what we assume, but we need to prove it. And the reality is often that this is not the case. There might be particular lipid that is, somehow not making it into the vesicles. So you have mixed 3 lipids, but in the end, the vesicles contain only 2. The third one is excluded. That's now an extreme case, very rare. But at least the exact ratio that you have mixed might not end up in the ratio of this. And so you need to prove this. So what you can do is, again, you take these here proteoliposomes that you have formed, and you can do this at different steps also here if you want. And you extract the lipids. And then you check. For instance, here we make liposomes containing basically here phosphatidylserine and phosphatidylcholine. So we used basically only phosphatidylcholine, and then we added more and more PS. And the hope is the more PS you add, the more the vesicle should contain on this new lipid, right? So in the theory, we would expect, okay, those should contain 10% of the lipid should be phosphatidylserine, 20%, 30%. And you see already, when you measure it, it's not the case. We have added 30%, but we only end up with 20% in the vesicle. Yeah? And what you do is basically, this is this lipid TLC, thin layer chromatography. You spot your lipid extracts, and then you separate basically the different lipids. And here you see, for instance, in the starting mix, you also detect here the detergent. In this case, it's oxyglucoside, also a detergent often used. and it's here present, but here it's gone. So you actually show by this technique also that you have been able to remove your detergent, right? Okay, so the take-home message is we can remove membrane proteins from membranes, but we need to put them back if we want to analyze them, to study them into so-called membrane mimics, for instance, in vesicles. To do so, we need a two-step procedure. We need the solubilization of a membrane with detergent, and we need the reconstitution step. The reconstitution step, it's not well defined. So you get vesicles that are empty, you get vesicles that contain few molecules of proteins or many. We cannot really control the way the proteins are inserted. They might be right side out, wrong side out. And when we make this type of reconstitution, we cannot guarantee that the vesicles really contain the starting lipid composition that we basically intended to have. Yeah? So that's important. So whenever you get samples from biochemists, be critical. Yeah? So Tripta is always asking me, Thomas, how much protein is in your sample? What is the protein-lipid ratio? What is the orientation of your protein? Because she would need all this information if she later wants to make conclusions out of the observations that she is, for instance, observing them in a microscope, right? Otherwise, it will be very hard. So we can make assumptions, but if we really want to understand, you would always also ask the biochemist to give not only assumptions, but maybe proofs as much as possible. Okay, cool. Next point. I told you, I think yesterday, that of course you also can include here now molecules. That could be, for instance, sensors, dye molecules. So let's say you want to study here, for instance, a transporter that transports ions. You could insert here a type of sensor for these ions. pH sensor, if it would be, for instance, a transporter that moves protons, right? But in this process, it's actually very tricky, because if you try this, so if you try basically here, type of pH indicator, you add your protein, you have your detergent basically, you have, for instance, preformed vesicles that you destabilize, or you just take lipids, and you take the indicator, and now you hope, whoop, everything in the end should be combined. Indicator should be here, lipids should be there, membrane protein should be there. That's the hope, but it doesn't work out often. Because what you see here during this formation of these vesicles, we lost here all the indicator. All the indicator was gone. Either was stick to the biobeads, or even in this gel filtration step, all the indicator was gone. So you get vesicles with your protein, but they are not filled with your sensor dye. Now you need to find maybe solutions to, to come around. And one solution is, for instance, that we decided to make these sensors linked to a lipid. Now the sensor is not water soluble, but rather likes to be with lipids. And this turned out to be actually very useful, because now your sensor stays associated with your vesicle formation. So that's basically here a type of pH sensor. Depending on the pH, it changes the fluorescence. In this case, the lower the pH, the higher is the fluorescence. So if the sensor would basically detect very acidic environment, it would be highly fluorescent. And you can, of course, here play around. And this is a type of, if you want to say, also synthetic biology, right? I mean, these type of molecules do not exist in nature, so you create them for your purpose. You combine a water-soluble sensor with, for instance, a lipid, and you can see that you can, for particular purposes, also modify the lipid. You can use a short-chain lipid or classical long-chain lipid. And that's something basically where you typically would work with chemists together. Now you need to go over here to the chemistry faculty and find a person that is somehow excited about these ideas, and then you say, okay, can you synthesize this? And then you can use this in your experiments. Okay, the other point that I would like to discuss with you is now this heterogeneity. You get basically vesicles in this procedure. We talked about having one protein, many proteins, no proteins. But what you can do if you take, for instance, type of large unilamellar vesicles or SOVs, you can immobilize them on a surface. Yeah. How we do this, we can maybe discuss later in the tutorials, but you can immobilize them, and then you can watch them under the microscope. Now, in this case, these vesicles, I told you, are very small. So you don't see really the vesicle, but if you have a fluorophore, you can see the fluorescence. So each dot is a vesicle. And that basically allows you now, if you have a fluorescent tag on your protein, if you have a fluorescent marker inside, you see the vesicle in one channel, in one fluorescent channel, and you can look whether there's a protein in or not. And on the intensity of the fluorescence of the protein, you can also determine how many molecules of protein you have. And actually, you have now here hundreds of little experiments. If you, for instance, here add a substance, you can check a vesicle without the protein, there should be no effect. A vesicle with a protein and the inhibitor acts, there should be an effect. A vesicle with many proteins should be also an effect, but maybe different. You see? So you can basically have one experiment, but you have each vesicle is actually an experiment, because each vesicle differs. So you get a lot of information. We call this single vesicle analysis, and that's now in the moment one of the techniques that is pretty much used by more and more groups, because this heterogeneity gives you in such a classical experiment an average. How could I explain it good? Let's say in average you are all very active, right? But that doesn't tell us, because some of you might be in this moment more active and others might be less active. And if I watch you over the whole day, you all will be different. And I will learn something basically in a way that what is common to all of you and what is different from all of you. And that gives a lot of information that is useful to understand molecular processes. Yeah. Okay. So the last point for this session is, how do we get now proteins into the GOVs? You are already able to make them. So that was basically yesterday a very good training, but these vesicles didn't contain yet proteins. Now you would like to make these vesicles with protein. Okay. The reason for that we motivated already. No? We can do with these giant vesicles a lot of nice experiments just based on fluorescent microscopy. So we could understand, for instance, the localization of membrane proteins, whether they are associated with domains or not. We can basically the association of proteins with membranes, the interactions with membranes, the effect of membrane or of, let's say, proteins that bind to the membrane on their curvature, on their on budding and deformation. And we can study, of course, transporters if we, for instance, use tricks like the sensors, biosensors that we insert into the vesicles. Okay, so electroformation you have seen yesterday, and gel-assisted swelling maybe also. At least you have discussed this maybe during the session. No? So what you use now, it's not basically the lipids or liposomes. You use your preformed proteoliposomes. And then you remove water, very careful, and you add water. And then a miracle happens. If you basically, under those conditions, use an electric field, you get GOVs containing protein. You can also do this with the gel-assisted swelling. So you put basically your vesicles, your proteoliposomes, in a type of gel, hydrogel, where they spontaneously start to fuse and to make these giant vesicles. It sounds easier than it is, because what are the problems? Dehydration. Very dangerous condition. Dehydration. No one, I have my bottle of water here. So life is based on water. You don't want to be dehydrated, and the protein also doesn't want to be dehydrated. So if you dehydrate too much, you get wonderful vesicles, even with protein. But the protein might be partially defolded, partially affected, unfunctional. And this we want to prevent. So it's again, it's almost like the Chinese would say the yin and yang. No? So to find the right moment, not to overdo it, but do it in a way that's good enough to get these giant vesicles. Okay, there's two more, charge-mediated fusion. The trick is here, you basically make proteoliposomes where part of the lipids are charged. Now you see that we use here lipids that are artificial. Part of the lipids is positively charged. This is nothing natural. That's something you need to buy these lipids or you need to let them synthesize, right? But you can basically then make your GUV. You have heard there's uncharged lipids, negatively charged lipids, and of course they would attract. And that means that brings them together. And eventually the vesicles fuse and you get your protein in. Or you can use these giant vesicles, as we discussed before, and start to destabilize them by detergent and add your protein from the detergent side directly. Then you need to remove the detergent, that the membrane comes tight again, and hopefully your protein GUV is in. So here's just one example. Rashani, I think you discussed this already yesterday a little bit, no? This type of gel formation where you use the hydrogel. So that would be basically you have your proteoliposome, you put it in a type of polymer hydrogel that also keeps a lot of water. And then basically these proteoliposomes start to interact, starts to fuse, starts to form these giant vesicles, and then you see under the microscope here the vesicle, and when you look in the fluorescent mode, and in this case the protein is carrying a fluorophore, has been modified with a fluorophore that shines nicely red. So now you have your protein inserted. And that's the start where you can then do a lot of experiments. You can, for instance, ask, does this protein now change the membrane properties, the membrane rigidity that we discussed with Abhishek, basically? Now, what would be the impact on stretching and bending when such a protein is now present? Yeah. So that's basically shortly the summary. I think I summarized a little bit in between already so that we can basically here finish on this moment. And I thank you very much for your attention, and we can go for the break. And